Thanks so much. I am John Junk. I am the, as somebody referred to me with the new name, geriatric. I just finished 50 years of college and university teaching this spring, and so, you're all off to a good start. As one of my colleagues says, she works for a fossil. That meant me. But I'm delighted to be here. There's a lot of people. I've looked for a long time forward to meeting you. We've missed one another at like 3 meetings and so on. I work with your colleague, Joe Ellis Monahan, and so on. And so please shout out hello. But I decided today that from a biologist, I'm split in 3. I like the poet James Cavanaugh says he envy the schizophrenic. They're only split in 2. I'm split between biology, mathematics, and education. And of course, none of the 3 departments at my university think I'm any one of those. You always must belong to the other one, kind of thing. But luckily I had an interdisciplinary institute where we literally try to bring them together. So I'm going to be an unabashed apologist today for the role of graph theory in biology. I really feel it has been so overlooked. As my friend Jim Keener at Utah says, that all of mathematical biology is partial differential equations. We with this workshop would like to disabuse them of that kind of notion, kind of thing. And partly I argue for several reasons. One is that it's highly visual, and I think it relates to many of our biological colleagues. And it also reinforces to biologists that many of the questions that they ask lend themselves to mathematical analysis. And many of these questions are simply binary. You're the child of whom? You're the descendant of whom? Who ate whom? Substrate, product, etc. That these kinds of binary relationships are many of the kinds of questions that biologists ask for. And so that when they produce something like a phylogenetic tree, a pedigree, a metabolic pathway, a cell signaling pathway, they already have done mathematics. They've done a generalization, an abstraction. Surely when we do a food web, it's not individual organisms. We're talking about whole taxonomic groups that we're dealing with that. So they have done a level of generalization and abstraction. And that really that these are formal mathematical objects, that they really are in graphs in the sense of graph theory, and that we even share the common language, trees, networks, paths, etc. kind of thing. And thirdly, that we have a different expectation. So this graph is derivative from 1966 work of Richard Levins, who argued that really the triangle usually of modeling had been due to physics, that they were interested in precision and accuracy and prediction to 10 decimal places. But in biology, we're interested, does it relate across a broad phylogenetic spectrum? Does it relate across different causal mechanisms, different spatial and temporal kind of scales? And I want to reinforce those kinds of relationships that very frequently our criteria for models in mathematical biology are very different than those in typical kind of physics kind of things. And think of it, we don't get to do experiments in zero gravity or in situations with frictionless surfaces. We're dealing with very complex ecosystems and so on kind of thing, and we'd still like to know something about that. And Mary Williams, back in 1977, said, since parts of future biomathematics which involve further development along directions already prominent can be extrapolated from other papers in the volume, the purpose of this paper is to investigate less obvious and therefore more speculative directions. In particular, I intend to show reason to suspect that the laws of biology are of a different mathematical form than laws of physics, and that therefore the mathematical models of the future of biology will be radically different from the models used in physics. And she then goes on to cast some doubt. She ends up by saying maybe graph theory might be the new language, that graph theory is biologists' calculus in the same way that calculus was physics' tool. So when we look at a typical kind of graph theory kind of textbook, that there is often more ties to applied problems rather than a typical kind of math book, this being one of them. And if very frequently, though, where are the examples? As usual, they usually come from physics, engineering, business kinds of things. And so what I began doing is, well, why can't we look in terms of graph theory with similar kinds of things with biology? And here's some of the typical kinds of graphs that you'll see flash by you today. And I'll warn you that you'll only see flashes kind of thing. But to get some kind of sense, they'll be familiar. And when I saw this essay that had broken out ways of thinking about visualizing information, it actually ended up being the way that I've structured today's talk, that let's start in a one-dimensional world and then move to 2 and 3, etc., and finish off with something with trees and networks kind of thing. So this is the organization of my talk. I'm simply going to take you from 1 to 2 to 3 dimensions, and then let's go to fractal dimensions, and then let's deal with 4 kinds of graphs, trees, networks, paths, circuits, and bipartite graphs. So if we start off with one-dimensional kind of graphs, partly I would argue that much of the 20th century was focused in biology on a one-dimensional journey. Sequencing proteins, sequencing nucleic acids, that it was very frequently driven by these kind of very strong kind of projects. It turned out, though, that when you did the math, that the math lent itself, as you can see in chapter 1 of the Yellow Book, that to a variety of other problems, including some current systems biology and food webs kind of thing. And partly I've been a software designer throughout my career, not really a programmer, but an architect of many things, and so we have some software on these kinds of things as well. So, and partly I want to also make the point that biologists were the motivation for the mathematics. So Seymour Benzer rewrote biology because prior to his work with viruses of bacteria, the gene was a unit of function, a unit of recombination, and a unit of variation, mutation. And what he showed was when you could grow up 1 billion virus particles per milliliter overnight, that you could go and do what he called the fine structure of the gene and literally map that recombination was between adjacent nucleotides in DNA, that you could have a mutation as small as an individual nucleotide, that instead of 1 gene, 1 enzyme, he had 1 cistron, 1 polypeptide. So think of hemoglobin, you have an alpha and a beta, so you have 2 of each to make hemoglobin. And similarly, this occurred in this viral kind of system. And these ons, mutons, recons, cistrons, have operated in biology ever since, and now we have metabolons and operons, etc., all of these kinds of things, since this kind of language that he invented. But he literally used deletions to figure out intervals and then map the points to it. And in his 2 PNAS papers, literally then the graph theorists began to do it. And we review some of this history in the first chapter of Ryan's book, kind of thing, for doing it. And I was most struck, I was reading literally a translation of a Russian article in 1960s where you simply could do partially linear ordered sets and do this, but we can do the same thing with kind of graph theory and so on. So taking on problems like sequencing, you take a long sequence, you cut it up into fragments. If you cut it in different ways, you're looking for overlaps. And so really the binary information here is, does one fragment overlap another fragment? And from that, can we assemble them into a whole sequence? And it was literally called the middle level problem of the human genome, that you had millions of these little fragments that you could sequence, say 300 to 1,000 nucleotides long. But if you're trying to do 6 billion base pairs, you have a few things to put together. And so how is it you do that kind of thing? And so what was wonderful, the biology textbook had a very ad hoc approach, and then you could go through some formal graph theory, some set theoretic, and then also some basic algebra in terms of rearranging matrices with consecutive ones. And so my students, I brought back a PNAS article, and it had this kind of figure on a yeast gene expression pathway. And my students who were used to solving problems in my genetics course said, hey, they haven't finished. And so here was taking that same data and using two of our software to reorganize and to look at patterns within. And so you might call them motifs, but the squares on the main diagonal or the fully connected colored sets over to the right in our software biographer represent maximal subclicks within the data, and the linear order along the main diagonal, or that you could take the order on the circle, literally represented the relationships between them. And so we would see the same pattern that you saw here in the lower left. And so doing a variety of these, you can see a little bit of the graph theory laying out the multiple steps and so on, some of the proofs that are involved. But how do you take complex data like the left and represent them in visualizations such as on the right, that think of the right as basically a metabolic, a mitotic cycle, and that if you rearranged it in this circular array of your graph, that you can see that there's blue and light blue and red and yellow and green and so on, purple, that arranged around a clock face, that you get some sense that if we were going from G0 to G1 to S1 to a mitotic division in a cell cycle, that this is like a clock. And so partly I argue that also even just the visualization of data is crucially important in terms of doing this kind of work. So it turned out, though, that the same mathematics of doing a one-dimensional array of a set of segments along a line came out in 1976 in a book called Food Webs and Niche Space by Joel Cohn, a Little Princeton University Press monograph. So you take a food web, and instead of looking at who eats whom, what you're interested in is what Dr. Seuss says. And new is the name is a nutch that lives in a small niche, but some other nutch would like your niche very much, and so nutches without niches will snitch one another's niche, okay? So, you know, so what you really want to look at is you want to look at the competition graph of which two predators are competing for the same prey, okay? That's my food. No, you know, you can't have it, right, okay? And so you go to this, and amazingly, what Cohn showed is that you could arrange the predators up here in red and blue along segments of a line, that in this complex data, so that here's the food web, there were only two intervals of this community in one-dimensional space. What we did in additionally then to extend it is these are the prey down below, and so you can see some of the blues are generalists. They're eating multiple niches of prey down here. But you get a different sense. Very frequently people will ask about food web, what if we remove one particular species? Who's it going to affect? If you go to this two-dimensional kind of display kind of thing, you get to see that a little bit easier. And then we went further to take some of the energy flow through this kind of system. And so what we did is we organized it in 3 dimensions. So the one dimension is the community of niches of predators competing with predators. The second dimension was the trophic level, so top predators and producers, et cetera, kind of thing. And then thirdly, what's the energy on it? And so partly what we're trying to do is to take something that simply looks like a cartoon in a textbook, like a food web, but then let's do some graph theory, and then let's think about multiple visualizations of going to 2 dimensions where we understand trophic levels as well as the community organization of intervals. And thirdly, let's look at, say, either energy or biomass flow through that kind of ecosystem. And again, visualization helps. So one of the things of taking the predator-predator matrix and displaying it here is with what's called a spring model layout of a graph. And here you can already see kind of the 4 intervals here of the purple, yellow, blue, and red in terms of it. And so by springing them out, you already see kind of these maximally clicked subsets and what the linear order relationship is amongst them. And recently, a Taiwanese computer scientist dealing with PQ and PC trees came up with an algorithm. We had been working with a heuristic in the past for doing it, and we've been implementing this with my Thai colleagues in Bangkok, kind of thing. If we move to 2 dimensions, that all of a sudden now we're dealing with planar graphs. And one of the delights of being here is I finally get to meet some colleagues who've been dealing with some of these problems like RNA folding and so on for a long time, kind of thing. But I in particular have been looking at some things that, again, biologists preceded the mathematicians, that they were looking at simply these vertices, edges, and faces of polygonal tessellations and asking questions about them. And then in 1908, Voronoi in Kiev, and in 1934, again, Delaney in Kiev came out with geometric, computational geometry, and kind of topological dual of these things. But so you'll be seeing here, and so Deborah and Jeff Knisley have done beautiful work on RNA folding. I love, these are just three of the two-dimensional kind of displays. Probably to most of you, you're familiar with the left-hand one, which is frequently called airports. So think of it as, you know, these kind of pods at an airport. The upper right is one of my favorites from Israel, Ruth Nusinoff, where they're called Nusinoff circles. And then to the right is one called domes. There's a fourth one called mountains. They probably know more two-dimensional displays than I know, but these are just three of my favorite. And so here's one of their recent articles with Robin and with Matt, who have also worked on this kind of problem. And so I thought I'd make a shout out to you. But there's a variety of other kinds of things that we're interested in with molecules, secondary structures in a variety of kinds of ways, and how do we look at these kinds of things. And so HP lattices, hydrophobic polar kind of lattices, or that looking at what are called the helical wheels by Klotz and so on kind of thing. But I've been particularly interested, I do a lot of microscopy in looking at these kind of tissues where we have these very nice polygonal tessellations. And this was Voronoi and Delaney that in 1908 and 1934. The construction of the nearest neighbor polygons, so basically all you're doing is the perpendicular bisector between two adjacent points. And you get these nice trigonal kind of areas where that is the center of a circumcircle that will go with equal radii each to each of the neighboring generator points. The nice thing about its topological dual, where you're connecting the points in the center of each of these polygons, is that's the natural thing to measure. What is the distance between you and your neighbor, etc. But it also is nice that very frequently, think of it if you had rain gauges out and you're constructing the average rainfall for each area, so on, but one rain gauge is covered with a leaf and you're missing a piece of data. You can just snip that and just do the local reconstruction. And so working back and forth between the topological dual and the geometric works very nice. So we have software that we design, again, with my Thai colleagues, that we simply can import any biological image, and then we can do some of the computational geometry of the Voronoi polygons, again, the Delaney duals, that we're only analyzing within what's called the convex hull. So we don't take any of these polygons that go out to infinity on the periphery. And then we have some graph theory in terms of various kinds of spanning trees and neighborhood graphs and so on. Some spatial statistics in terms of whether you're random, whether you're clustered, or whether you're over dispersed. And then some visual displays, in this case just the number of sides of polygons of the different types and so on. And there's a few other things in the lower right. We look for some technical things like Pitaway violations and so on, whether a Delaney triangulation edge crosses over two Voronoi edges or not only one kind of thing and so on. Yep.
Analysis there, or is somebody keying in where they think?
Yeah, you're keying it in, and so so very frequently you're simply clicking on nuclei or florets of a particular plant and so on. We have not been able to do some automatic thresholding and so on, but if you're a good enough computer scientist to help us out, we're interested in going in some of those kind of directions. But and so we last September had the copy of Microscopy Today, where we actually were studying these little fossil microorganisms called radiolaria. Basically, think of it as an amoeba in a glass house. And so they could stand up to X-ray tomography and build the whole 3D. And then on the lower right, a woman saw our cover article, and she was an artist who has a fairly good math background, and we sent her our whole 3D files, and she did laser sintered glass. And since these organisms are made out of glass, it's amazing. For the first time, I can do 3D printing, and so I can hold one of these baseball size in my hand. But now with these laser sintered glass, it's a beautiful piece of glasswork, and it's more like what the organism might look at, because we have glass, you know, kind of thing. And so kind of fun in terms of doing this. But we also can ask important questions. So here was a movie of embryogenesis in Drosophila, and we had 5 generations of synchronized mitotic indices. And we began looking at that kind of data and so on, and seeing whether it fit a variety of simple kind of relationships and so on. But my point was that in doing this kind of work, I became fascinated. Isao Honda had an article in the 70s in Journal of Theoretical Biology, and I read it. And it was like, you know, we biologists get accused of doing postage stamp collecting. And so all of a sudden I began looking at all the images from a wide phylogenetic kind of diversity. And so one of the things is here you can see that across the phylogenetic tree that these patterns reoccur time and again. And so one of my basic assumptions for building mathematical models in education is that my biology colleagues, that if the mathematics really deals with that broad phylogenetic survey, different spatial dimensions, so we're dealing with nanostructures that fold in 4 nanoseconds, to things that canopies that we might be looking at a kilometer scale kind of aerial photograph kind of thing. That if we're looking at some tropical rainforest and a tree canopy, it might have taken 300 years to develop that mature canopy that you're looking at, versus these kind of nanoseconds of folding of some 3-dimensional structure kind of thing. So we have things from, you know, nanoseconds to microseconds to seconds to minutes to hours to days to years to centuries in terms of thinking about it. And they're caused by different kind of mechanisms. So that you, in some cases, it's a, think about this fish swims out and spits out sand to create a boundary halfway between your neighbor fish, you know, kind of thing. In another case, you're growing until you're touching. In other cases, you're diffusing. In other cases, you're packing things, okay? So you have different causal mechanisms, but the same mathematics is fitting across all of these. And also, since you're dealing with point-point interactions, we can look at simple things. So this was my colleague's data on red-winged blackbird nests that were successful and unsuccessful, and he thought there was a clustering, but we simply did the simple binomial theorem, p squared plus 2pq plus q squared. So do you have successful, 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 unsuccessful, or unsuccessful, unsuccessful edges? And you can just count them and look at, and did you have a random distribution or not? And he was quite disabused that it was random, that you couldn't reject randomness in this kind of case. Because we want to impose pattern when we're out there. And you know, you're out there in the field and you think you've seen something. And so the hardest job in my life was working as a statistician at a veterinary administration hospital with all these physicians who were sure they had something significant, and you do the statistics and disabuse them of that, and they didn't like you forever, you know, kind of thing. So, you know, so let's go to 3 dimensions, and we'll go from polygons to polyhedra and polytopes and so on, kind of stuff. And here was a beautiful image of a Drosophila egg emerging from the ovipositor of a female fly. And the author had made some claims that it was all covered with hexagons. There was a similar story about radiolaria, and Darcy Wentworth-Thompson quoted that you know, that showed biology was better than God, and then the mathematician said no, it would have caught God in you know, an inconsistency kind of thing. But with a soccer ball theorem that some of you may remember, that the soccer balls in South Africa with the World Cup were too hard. They had put too many hexagons on them, and they'd gone to a finer grain and got a harder ball that didn't behave the same way that most people were used to playing with it. But here was this classic Euler formula: vertices plus faces minus edges is equal to 2. And with that, you could prove that you cannot cover a surface no matter how you deform your hexagons with all hexagons. And so I just highlighted here some non-hexagons on the surface of that Drosophila egg. But, you know, with wonderful language and purple prose, they went on and said this ghostly mess of hexagonal lines. And, you know, yeah, hey, so many people have done it. There's an animal behavior paper on the fish territories called the hexagonal territories. You look at it, there's pentagons, there's heptagons, you know, kind of thing. And so this is what you would expect. And I also argue a great deal that it's important to go through some of these simple proofs, like the simple disproof that you can not cover, you know, a polytope with all hexagons kind of stuff, and other kinds of things. And I think it's crucial. Some beautiful stuff in terms of viral capsids and 3D structures. Casper Klug won the Nobel Prize. I did an article in the 90s called 10 Equations That Change Biology, and many people critiqued me for the 10, and this is certainly one that I would have added if I extended to it. Beautiful piece of work in terms of being able to do it. And recently a woman in northern England, Tuareg, has done stuff to extend it, that there were some viral capsids that didn't work, and she had to do Penrose quasi-crystals in order to do this. But up at the top, here's the typical thing of T numbers of doing these icosahedral kind of viral capsids and so on kind of stuff. One of my favorite things is, how does a cell hold its shape in three-dimensional space? So my friend Greg worked for NASA, as I did, and how is it that when you go into outer space and you don't have gravity, that your cells can still hold their shape? Well, an old notion called tensegrity, that if you think of microtubules as kind of rigid rods, and if you think about, I'm blanking, but these G protein kind of things, that intermediate filaments as kind of rubberized kinds of things, with these kind of struts and tendons, that you can create these kinds of things. So in the upper left is Donald Ingbert's Scientific American cover in terms of the architecture of the cell. But, you know, this allows you to build a structure that you can fold flat, a tensegrity structure, and make a tower that can act as a bridge that you can go across a ravine. And so literally used, for instance, in Antarctica, where they hit these huge ice crevasses and so on, or sometimes hikers in mountainous kind of areas kind of thing. And so that in one sense, these are completely collapsible, but in another sense, they can maintain a three-dimensional structure. And so there's nice graph theoretic kind of rules of when do you have a rigid structure, when do you have a flexible structure, and so on kind of thing. We also, in terms of 3 dimensions with proteins, that we're interested when you fold in space, which amino acid side chain is next to this one and next to that one. And so instead of building Delaney triangles, here we do 4, and we do a tetrahedron. And so you do Delaney tetrahedra as the topological dual in this kind of case. And you can also then do these space-filling foams that are like Voronoi 3dB kind of polytopes in this kind of structure. And that's even one of the kind of visualization things. In the upper right is another one called ribbons kind of stuff. So let's go to fractal dimensions. And one of the things about fractals is that here we have self-similarity operating, and that with very simple rules. And so sending students out on campus, and so Jeff nicely at East Tennessee State literally had his students go out and do it. And I can't remember, Jeff, you built broccoli, and what were some of the other things that your students?
Well, broccoli, oak trees, bananas. Of course, I take an example of furnace. A lot of them have ground up furnace.
So Barnsley's ferns are some of the most famous fractals ever kind of thing. But so we just give them a spreadsheet. We ask them to kind of measure, pick out a little twig that looks like the whole tree, and then look at the branching angle, look at the distances between nodes, to look at the phylotactic angle. So as you go up, how many kind of branches do you go up before there's another branch coming off at the same angle from the central thing as the one that you're on, and getting a phylotactic angle. So you've got diameters, you've got distances, you've got two kinds of angles, and you've got the total number of bifurcations that you're doing it. And put that in, and in the lower right, that was a little tree outside our lab in Bangkok that we did with this kind of thing. And that it's important that students understand with the self-similarity, but also we then have them measure the fractal geometry with a variety of tools, these kind of log-log plots of log of the size of a box versus the number of boxes to cover a particular kind of image. And in our book, Microbes Count, where we have essays on doing this kind of thing. But you begin to understand some of the morphospace of bacterial colonies that when you're not growing them on, rich kind of wet, warm media, but, think of a bacterium out in nature that it might be on concrete, it might be real slow growing, it might be starved, etc. And some of the most fractal kind of dimensions that we get, and wonderful work from Israel with these images in the upper left, and given us permission to reproduce them and so on. And so they're very diffusion limited, and so they're like Ulam trees in graph theory that you're self-avoiding kind of walks in terms of producing these kinds of structures. You don't want to go where your colleagues have already gone and sampled the kind of space kind of thing. He literally got the centerfold of Scientific American with these beautiful bacterial kind of structures. And so many times our colleagues think bacteria are yucky and so on, but here are these absolutely, to me, gorgeous kind of colonies that have pseudocolored and so on. But it has real practical dimensions. So for instance, in cancer, here's a hairy cell leukemia in the upper top compared to a similar kind of cell. And think of it when the area is staying constant, but the perimeter or the volume staying constant and the surface area is going to infinity. The communication with your neighbors is completely changed. Think of a nerve cell with extraordinary number of dendrites versus being simply a cubic or a spherical kind of cell. That you interact with your neighbors in a very different way when you have that kind of membrane contact and so on. So doing the fractal dimension of a ruffled edge like this can frequently even have prognostic, diagnostic, treatment kind of impact in terms of understanding something from a cellular pathology point of view. So let's go to trees. And so as an evolutionary biologist, phylogenetic trees are crucially important, but taught genetics with pedigrees, and as long as you don't have any inbreeding, consanguinity, that they can be very tree-like, but also in terms of embryology, teaching developmental biology, the fate maps are similar kind of thing. And so, here you have some of these kind of phylogenetic trees that, I love the diversity of these kinds of things. And one of the things that's very hard for students to represent is that if we look at the tree of life, in the upper left-hand corner, it says, "You are here." We're a bunch of Johnny-come-latelys on the history of life that, in the last few seconds before midnight on a 24-hour clock, we appeared. And somehow, Galileo gets a bad time for kind of not having the Earth being the center of the universe, but for us not to be the center of the universe of a phylogenetic tree is also disturbing. But I think it's fun that, here people with kids and so on do these kinds of things, and doing the tree of life on a tree. I mean, how much fun can that be? But a very simple kind of graph theory in terms of doing this kind of work. Walter Fitch, I wish I had done my PhD with him, really working with Emmanuel Margoliash, who worked at Abbott Labs outside of Chicago in terms of constructing these kind of things, that if we take sequences and count how many differences they are from one another, we can get a distance matrix. and from the distance matrix. If we just have 3 sequences, we can do a completely deterministic 3 equations, 3 unknowns to determine the lengths of the branches, but you can't go on beyond that, so what they would do is collapse branches and produce an algorithm. And it's amazing that there's so many new algorithms, but they're frequently derivative of this 1960s kind of work. And one of the things that we focus on is that for every tree that you actually test it. So we have software in the upper left, EvolveSeek, where it generate problems that students can solve and come up with their own algorithm to build trees. But then after they built a tree, that you would actually test it. And so I'm going to show you this classic case that changed dentistry forever. So there was a dentist in Florida that had infected patients, and the question was the dentist the source. And so here's a tree with the dentist and the patients, and then other members of the community with HIV. And what you try to do is that if we just take the dentist and the patient versus 2 members of the community, with 4 what we called operational taxonomic units, there's 3 distinct trees. And what we try and do is compute what's called a split decomposition index of simply taking, in this case, these 2 kind of boat configuration distances minus the 2 kind of arrowheads. And what we're left with is 2 copies of the middle branch, and that is your split. And so how much have we changed A and B from G and D in this case? And literally with this distance matrix, the only one of the 3 topologies that fit was the dentist with the patients that were infected. You go to a dentist today, and most of you probably are too young to remember, but they used to dig in your mouth with their hands. Okay? And now they wear gloves. They ask whether you're latex sensitive and which kind of glove they have to go in. Sometimes they're wearing a dental dam over their mouth, etc., kind of thing. That literally here was a case where evolution and mathematics and David Hillis and other mathematical biologists literally changed practice. Now it's been called into some question and so on, but nonetheless it holds up that these phylogenetic tests work in court. And so a physician who injected his former girlfriend with HIV literally got convicted of attempted murder, in this kind of case, based on the phylogenetic analysis of the sequences of her virus versus the kind of viruses that were in stocks in his lab, kind of stuff. So here it changed forensics and the law. And so we live in a culture that's kind of anti-evolution and so on, and yet we have entered into kind of forensic practice, and it's because of this kinds of mathematics in doing it. Here's simply just a case in terms of development of which cells come from which and so on. We only know this for one animal. Does anybody know which animal it is? It's got a beautiful name. Caenorhabditis elegans. It's a little glass worm with 959 cells in the adult, and it is the only animal where we can go from a single fertilized egg to every cell in the adult. It is tremendous that we have this model organism for doing that kind of thing. And, here's a similar kind of thing of doing that kind of work. And you can see that if we try to look at us, this is my friend Scott Gilbert at Swarthmore, just recently retired, that we're really only able to do it almost at the tissue level. We're not able to do it as cell-by-cell kind of level. So we as human beings, we don't know even our own origin from a single fertilized egg. Or I try to talk to students that they need to understand phylogenetically you're all chimeras because your mitochondria are related to a purple photosynthetic bacteria and have a different genetic code than your nucleus. You are a bacterial colony, probably an archaeobacteria and a purple photosynthetic bacteria in terms of your history. They don't think of themselves as bacteria, but as George Bernard Shaw said, a touch of Darwin makes the whole world kin. So, here we are kind of thing. But I think it's important that we understand ontogenetically, genetically, phylogenetically, that these graphs really define who we are as an individual, as a member of a family, and as a member of our evolutionary history. That this simple kind of graph theory is crucial to doing all of this kind of work. So we worked on called Pedigree Construction Kit, and I worked with a colleague in Egypt, and they have 33% first cousin marriage closer in Alexandria and Cairo, 75% up the Nile, and 100% on the oasis villages. The consanguinity is so high in Egypt that they see Mendelian autosomal disorders higher than any other place in the world. It's basically a genetic laboratory. And literally, at a crucial time, she had to smuggle out her notebooks for me to work with in developing software for her so that literally they could see the relationships of recurrence and inbreeding and counting these kind of calculations and so on and so on. But it's also, tree building is a wonderful problem that I throw out to the mathematicians, computer scientists, because it's both overdetermined and underdetermined at the same time. So we have more data than we have distances. It's overdetermined. But the tree topologies, if you have 22 sequences, you have over a mole of trees, over Avogadro's number of trees. So very frequently, some centers will literally have a say, do not submit a job size bigger than this. Now, it turns out we can do hundreds, and that's because the algorithms pick up on the evolutionary homology and speed it up and so on. But for the most part, very frequently, students will be in a lab and say, oh, the supercomputer must be down in San Diego. No, the supercomputer isn't down. You just submitted too large of a file to be done in a finite period of time. And so partly for me, I go through a proof of counting the trees and topologies and the distances and so on, simply that they begin to understand not only do they have to come up for their own algorithm for building trees, but that by doing a kind of proof of the combinatorial explosions that are going on here, that you understand what are some of the ramifications for doing the computational analysis of your data. And we live in a world of big data, and how do we prepare our students for dealing with big data? So if we go to networks, everybody these days is dealing with it. A procedure of this workshop, we for 3 years had been bugging NIMBioS that we wanted to do a workshop on graph theory and networks, and for 2 years they turned us down, and finally they said, well, usually we want 24 participants, but if you can get 12, we'll let you run the workshop. We held the record for many years that we had over 90 applications before they shut down the admissions to it. Matt, you were there, kind of thing, and amazingly we accepted 35 to keep them happy, and thanks to Raina and Matt for organizing kind of a follow-on, because they've been bugging us to do it again, and thank you all for being here and seeing that this kind of work's important. So, you know, one of the things I want to emphasize with networks is, again, the power and the importance of visualization of patterns and data. And so here's our leader, Fearless, that she has worked heavily on this, and I think, Matt, you're going to tell us about new work you've been doing with Boolean networks as well, kind of stuff, that these things are crucial in this kind of area. This is one of my favorite kind of illustrations from Cytoscape, graph visualization kinds of tools, that coloring and laying out these kind of patterns are crucially important. Drew, are you going to be able to talk about your software here using NetLogo or not? Sometime, I presume you'll show some of this? Okay, but he was always willing to show it, you know, kind of stuff. Okay, great. So, you know, there's a variety of wonderful kind of packages for doing this. This was our own software that's becoming a little dated. That what we wanted to make easy for people is to take their data into an Excel spreadsheet and then to be able to graph it and so on. We really found that with so many of our biology colleagues that the spreadsheet was the tool that we could get them to use. We couldn't get them to use Mathematica, as my colleague Tony Weinstein said. Just even the name intimidated most of my biology colleagues, you know, kind of thing. But they might have been doing a spreadsheet in the lab to collect data, or they might have been doing their grades in it, and so that we could get them to use a spreadsheet. One of my favorite tools for analyzing networks looks at a really small motif, a triangle. And these are just the 13 kind of relationships amongst 3 nodes in terms of directed graphs. And a technique that was published back in Science about 10 years ago called triad profile analysis. It's actually been used, one of my own most interesting cases was looking at student collaboration kind of thing, and whether they were more effective in surviving as effective learners by their collaboration kind of strategy. And it turns out in a very nice kind of study of looking at Arabidopsis thaliana is a model organism plant. I think most of you recognize Homo sapiens, but in the Miss Saccharomyces cerevisiae is one of the most common yeast. And so here you have 3 different kingdoms, plants, animals, and fungi, and that their use of these triads differs significantly kind of thing. And so the network of organization of our genes interacting is that even though phylogenetically we often have the very same genes, the networks that they're connected to might be more preferentially. So which genes are turned on when, how long are they turned on, et cetera, become really important things. It's not that we're so different in what we are, it's in that how we're used temporally, et cetera, and spatially in terms of these systems. So a whole variety of kind of networks, so looking at the spread of disease in terms of an outbreak graph with AIDS, or looking at metabolic pathways, kinds of stuff that looking at proteins at sites that co-vary. This is a kind of a household level of infection. I love Victoria Caliza's work in Italy, where she literally looked at the spread of SARS across the world, and here particularly in the United States, but she literally mapped out the airplane pathways, and she took only those flights that, say, had more than 100 passengers and used that to predict what was the spread of SARS around the world with really quite great faithful kindness. And this was just looking at, in China, the spread of SARS, you know, by taking hospital admissions and so on, kind of thing. And looking at other things, so looking at, you know, diseases of contemporary society like obesity and social network in terms of heart disease and so on, kind of thing. That these kinds of graphs now are very frequently crucially important for us to understand something about human disease. We'll hear more about neuroscience and the connections within the brain, and that if you think about these kind of really, that we've gone from much more simple models to much more multi-connected, that, you know, if you look at a model with, say, 39 simultaneous differential equations, how do you make sense of it? But I would argue that with graph theory, we have things, tools to make sense of some of these complex patterns. So, you know, the advertisement, better living through networks, you know, kind of thing. Pediatrics, where they actually changed the nursing model and did better, looking at human diseasomes or maps of the clustering of those things involved in various kinds of diseases and so on. And looking at polygenic, so where not one gene disorders, but many genes and the role of environment and so on, kind of thing. So, you know, one of the things becomes important is what are the properties of a network that are important? And one of the things that we've seen is that many of the real-world networks of biology are small-world networks, that there are some long-distance connections that connect little subgroups, kind of thing, that most have some clusters and some short lengths, and that there's some fragility in the edges that connect subgroups. And so very frequently the focus had been on hubs that are well connected, but looking at this, short ones. And so looking at whether it's random or scale-free or small world, that these things have been important in kernel networks and so on, and providing these kind of things. Also, one of the things in dealing with phylogeny is, again, a tree is a planar graph, but if we go to a multi-dimensional network of phylogenetic relationships, in many cases we can preserve the distances, and we then can ask how treed is the graph. And so one of the things by using software like splits trees and so on, that Daniel Hewson, the first author of this book, recently won $1 million prize from the National Security Administration, NSA, for solving a complex problem. So here is evolution solving a problem for cryptoanalysis kind of thing. So these things really make a difference for us to not always presume, let's shove it into a tree and that we can make some really interesting. And I'm very excited, and I'm literally hoping that Raina and others are going to help me try to understand some of the work of Gunnar Carlson at Stanford, who's been doing huge topological data reduction analyses and found important things, particularly with breast cancer, but developing whole new techniques of how do we prune these huge networks and find really significant subpatterns and so on. And this is actually developed a company for doing some of this kind of work. Last two groups, so paths and circuits. Most of you have probably heard of Eulerian and Hamiltonian kind of paths and so on. And so what happens, what's the difference between visiting every city once and only once versus traveling every road once and only once? So the visiting every city, the Hamiltonian problem. Here's one of my favorite cases, folding a viral capsid by self-assembly. It's called four-dimensional printing. Literally, you can buy a bottle. Unfortunately, the company hasn't lasted, but you shake up the pieces, and it's called four-dimensional printing, and you end up with a dodecahedron there. And they actually can do a left-handed and a right-handed dodecahedron. And so I'm trying to seek help from your colleague Jo Ellis Monahan, who has her rules for DNA self-assembly. How can we do these different icosahedra of different things? And I'm hoping she'll bail me out, kind of thing. So, but I've worked a lot on genetic codes as codes. So here's one from my colleague Rosemary Swanson, where you're only changing one of 6 bits. So if you encode each of the 4 bases with 2 bits, and you only change that, you can change all of these going around. And what she showed in the center was she's a X-ray crystallographer, which residues tended to be on the inside of proteins and which ones on the outside, which ones were small, which ones were large, that mutationally you were only tending to go within those kind of groups for the most part. And this is another kind of representation of that. Another Hamiltonian path is doing a Rook's tour on a chessboard, and there was this article in Bulletin Math Bio, and I had one of my students do a different kind of Hamiltonian path where we found that our physical chemical patterns were changing less on it. But the problem is there's so many Hamiltonian paths that we couldn't find what was the optimal one. So some computer scientists have some idea on how to approach that kind of thing. But Eulerian, where we walk over every walkway or every kind of street once and only once, or every bridge in the classical kind of Euler problem kind of thing. These are called Bordeaux codes rather than gray codes. And so I was very proud. I had taken the 16 dinucleotides and found all of the pathways to generate this kind of code. And I was presenting, and a coding theorist said, well, John, you discovered what De Bruijn became famous for, but after it was named after him, it found that a woman in the 19th century, Flynn de Saint Marie, had actually anticipated him by almost 100 years. So, there popped my bubble, kind of thing. But this was my genetic code of simply, with 64 nucleotides, I could have all 64 codons. And actually, at the time, I was working on a microcomputer with 24K. And so reducing the problem, I had a very pragmatic reason for doing it. And nobody today with terabytes can imagine doing that kind of work. But there are Haas diagrams and Boole lattices, et cetera, for doing this. But we can ask questions about error detection, error correction, and minimal mutations. But we also can do sequence assembly problems, both with Hamiltonian and De Bruijn. Some nice things with students for them to play mastermind and understand the difference between sequence and composition. We have some software for mimicking this kind of laboratory stuff. Huge metabolic pathways and so on kind of thing for doing this kind of thing. But really for people to understand that if you stand back from your metabolic pathway and just simply look at it topologically, do you have lines? Do you have circles? Do you have bifurcations? If you have a line, there's most likely going to be feedback inhibition, that something at the end goes back and inhibits something at the beginning. And if you have a circle, if you add a little bit, everything speeds up. If you take a little bit out, everything slows down. To me, that understanding the topology, that flux analysis, is different than the enzyme kinetics that we could look at an ordinary differential equation of each step, but understanding it. And Tim Komar and his colleagues have done some beautiful stuff at looking at multiple steps in terms of these paths and coming up with general rules and so on kind of thing, and looking at these subgraphs and so on. And finish offing with bipartite maps, because I give a talk says math saves lives, and I guess generally audience who are skeptical, about mathematics, and I say, which would you rather have? A brain biopsy or a CAT scan? Which? Yeah. For the most part, I think you'd prefer not to have them, cut out a piece of your skull and go in drilling and so on kind of thing. And it's 19th century mathematics called the radon transform. That is the Nobel Prize for literally doing this kind of visualization. Mathematics truly saves lives. Very frequently we tell our students, it's to keep you, calculus is a requirement for medical school to keep you out of medical school or something like that. And I think they really deserve a better answer than that, okay? And so that's why I think it's important to give these kind of things. So bipartite graphs are crucially, so in 2012 it didn't win the Nobel Prize for Medicine, but it won it for economics. And literally one of its application was literally, so here, applying math, saving lives, is the kidney donation problem. And how do we do this? And so at Stanford, they literally have a game online for doing this kind of kidney matching and figuring out an algorithm. And it's a huge economic as well as medical problem. This is the longest kidney donation chain. So you can't give your kidney to your kid, but you could give a kidney to somebody else's kid. And how do we do this kind of matching problem? It originally was called a marriage problem. It was published in 1962 in college mathematics, and it was frequently used for matching residents with their appointments after their residency in medical school. And here was one showing it worldwide to the Philippines and so on. But very frequently bipartite reactions are used, things like substrates and products. A wonderful piece of mathematics called the Hungarian matching algorithm, as well as the Gale-Shapley kind of algorithm. And so you can see this kind of demo on Mathematica and so on. But also with sequence alignment problems and genome alignments and so on. If we take, say, a host and a parasite, is the tree of the host the same as the parasite, or of the flower and the pollinators, in terms of if we're looking at coevolution kind of problems? And these are called tanglegrams. But one of the things been particularly interested in is in genomics, where we're looking about this problem called the pancake flipping problem. If you want to serve it all burnt side down kind of thing. And Bill Gates is most famous that as an undergraduate that he made a very slight improvement on this particular kind of problem in terms of it. In computer science, the greedy approach to doing this kind of thing. And so you're trying to say if you can recombine genes in terms of different orders and so on. And so our software, again with my Thai colleagues, pGraph, we can take about 200 bacterial genomes and to simply walk through and figure out the various kind of rearrangements to turn one into another. Get an evolutionary distance, we can actually produce a tree. But I love to do a shout out. Lori Heyer at Davidson College with her students literally solved this problem with genetic engineering, and they won an undergraduate competition with doing this kind of thing. But the question becomes, how do you turn one organism into another kind of thing? And very frequently, since these kind of rearrangements occur during tumorigenesis, that now that we can do DNA sequencing of individual cells over the course of tumorigenesis, we're actually building phylogenetic trees, and we're frequently looking at these major kind of rearrangements and so on kind of thing. So I want to simply close that biologists have already been producing in many of their labs these kind of visualizations. They frequently don't understand the mathematics or the computer science beyond it. We need your help. We need your help. And don't put it down because they don't understand it. They have real problems, and they have already done some abstractions and generalizations and idealizations and teaching about what they're doing. And so I ask you that frequently, as I've been a translator, marriage counselor my whole life, that how do I abstract and generalize my biologist and physician colleagues' problem, explain it to you as mathematicians, computer scientists, convince you not to just do your latest algorithm or your latest theorem, but to really pay attention to their problem. And then I have to interpret that what you did actually helped my colleagues and made a difference. But with that, I really thank you for this opportunity, and I really feel that we have extraordinarily interesting problems in biology that lend themselves to the kind of mathematics you're going to do here in this workshop. And I'm delighted that you would put up with this old fossil. Thanks so much, and thanks to my funders.